Rice is one of the main food crops that is liked by people of Burkina Faso, the country of honest people. The yield losses often observed in rice fields are not systematically linked to edaphic factors. There are also pests and diseases that pose a major threat to food security. Among the pathogens of rice crops is the bacterium Xanthomonas oryzae, Pathogen oryzae. This microbe is responsible for vascular bacterial wilt in rice, commonly known as bacterial wilt. It is a devastating disease well known in Asia and West Africa. In Burkina Faso, this pathogen devastated rice plots in Bargué in 1998 and 2004, resulting in losses ranging from 50 to 100%. To get rid of these pests, researchers from the Institute of Environment and Agricultural Research (INERA) are being called upon to create new varieties of rice such as TS2, FKR62N, and Orilax.
During our studies on bacterial wilt, we sought to understand the resistance of the collection of rice varieties developed by INERA against this disease. We realized that most of all varieties adapted to irrigated rice cultivation are susceptible to bacterial wilt. On the other hand, rice varieties adapted to rain-fed rice that have japonica base are resistant to the disease. Whereas, when we look at national rice production, irrigated rice production covers more than half of the national production. So, if we have to use rain-fed varieties in irrigated rice cultivation to resist the disease, we will not have the necessary yields, and the objectivity of covering our rice needs will not be covered.
The causative agents of vascular bacteriosis hijacks the rice sugar production system in its favor. As the bacteria becomes numerous, they clog the subconducting vessels, causing plants to wilt.
Our studies have made it possible to understand that rice uses sugar for its metabolism. That is to say, for its functioning, its development, its growth, rice produces sugar. And the bacteria also need sugar to develop. So, once a bacteria infects the rice plant, the bacteria will hijack the sugar produced by the rice to multiply. So, once a bacteria multiplies, it obstructs the subconducting vessels. That is to say, the rice will no longer be able to supply itself with water. The bacteria only benefits from the rice, and eventually the rice will wilt. That's why we talk about bacterial wilt.
It is therefore necessary to find varieties that are not only resistant to bacteriosis but also suitable for irrigated rice cultivation. Several techniques are available to researchers. It is possible to make crosses between susceptible and resistant rice varieties, but this classic crossing process is very long. Beyond these crosses, genome editing through CRISPR-Cas9 is the powerful tool that has made genome editing faster, more accurate, less expensive, and simpler to implement. The CRISPR-Cas9 system was identified by researchers Emmanuel Charpentier and Jennifer Donner, who are working on the Streptococcus. The discovery that earned them the Nobel Prize in Chemistry in 2020 was a highlight of the fact that CRISPR is a natural defense mechanism found in bacterial DNA, and Cas9, an enzyme, could be programmed to cut DNA in any location. The CRISPR-Cas9 genetic scissors are one of the major scientific advances of this century. It is a technology that has the power to revolutionize the society through its multiple applications in various fields, including agriculture.
Gene editing goes beyond GMOs. It takes the genome of a plant. It looks at what aspect he can correct to improve this plant. Everything that is made within the plant or animal organism is codified at the level of chromosomal DNA. So, It's just sequences that are there. So depending on certain sequences, it can cause certain abnormalities and insufficiency. Gene editing can work to correct what may be wrong and improve certain abilities of a plant level. It won't bring anything from the outside. So it's only a manipulation within the genome of the plant of interest.
In editing, there is no transgene. This means that we didn't take a fragment of a gene somewhere to insert it on the rice. The editing consisted just of modifying the sequence of the susceptibility gene, that is, the gene that produces sugar. This modification consisted of inserting or removing a few nucleotides, but there is no insertion of DNA fragments from elsewhere.
It means we know the sequence of the gene that make rice susceptible to a disease. Let us assume that this sequence is A, B, C, D to Z. When it is A, B, C, D to Z, rice is sensitive. On the other hand, if the last one is not there, the rice is resistant. So we can go into the rice and cut the Z. Rice is normal because we have seen that there is a form of rice that doesn't have a Z and that resists. So our rice, we want to maintain its characteristic but with resistance, just cut the Z. That is an addition. There can be the arrangement. If it is A, B, C, D, that makes the rice susceptible, while in other varieties, when it is A, C, B, D, the rice is resistant. We can then enter the DNA molecule and make replacement. That is genome editing.
C'est ça l'édition génomique.
The editing consisted of modifying the gene that produces sugar in rice. So when the bacteria comes, it can stimulate the production of sugar, but not enough to survive and grow, and suddenly the plant resists.
Security Level 2 Greenhouse. All measures are taken to prevent contact between rice plants and the human body. These provisions limit the dispersion of plant material. In this confined greenhouse, Dr. Issa Woni evaluates the rice edited under the supervision of the National Biodiversity Agency, ANB. It determines the agromorphological parameters of 3 generations of edited rice and tests the resistance of these lines to vascular bacteriosis.
When comparing unmodified parents and mutants, the difference is very clear. When inoculating the plants at the age of 45, 1 month later, the parents wither, while the mutants do not wither. Infected parents do not produce while there is production in mutants. So which means that the technology is interesting. The trial we are conducting is for scientific purposes. We want to know if the technology works. Is the technology effective? Does the technology adapt to our growing conditions? Varieties such as TS10 have been abandoned because of this disease. If the technology works in 1 or 2 years, we can reintroduce this variety into our cropping system. If the technology works, we can apply it to other crops other than rice, where we have genomic data on the plant and on the pathogen. There is, for example, cotton, which has an economic interest. There is cassava, which it takes an important place in production in Burkina Faso. Therefore, the technology, in addition to rice, can be used to improve the resistance of other crops to biotic factors.
The greenhouse results shows that, with exception of disease resistance, all edited lines are identical to unedited parents. On the basis of these satisfactory results in the greenhouse, the researcher obtained an authorization from the National Biodiversity Agency for evaluation in confined fields. This time, Dr. Woni is setting up an experimental site in Bama in Haut-Basse. This commune of Burkina Faso, known for its rice production, the research team will take necessary measures to limit the dispersion of these lines, particularly pollen grains, on any other variety of rice not concerned by the genome editing. The objective of the experiment is to evaluate the adaptability of the edited rice lines in real conditions and to ensure that in the fields these lines have the same agromorphological characteristics as the unedited parents.
Here we have two elementary plots. To my left, you have the variety IR64, and to my right, the variety mutated from IR64. When you look at the physiognomy or phenological stage of these two elementary plots, you will not notice any difference. In addition, when you take the IR64 rice plant with a mutated IR64 plant, there is no difference in terms of the number of tally. When you look at the maturity of flowering, semi-maturity cycle, you see that we have ears that are identical. Whenever agromorphological characteristics, there is no difference between the parent and the edited line. So, which means that the editing does not impact agromorphological parameters. So, here is the advantage of editing. It allows us to create a new character on an individual who has shortcomings that we want to improve.
Experiments in greenhouse and confined fields have shown that genome editing does not impact agromorphological parameters. Also, in confined greenhouse, tests have shown that edited rice is more resistant to vascular bacteriosis. What will then be the behavior of genetically edited rice in confined fields in the face of vascular bacteriosis?